Guess what? I'm back here to talk to you today about light and photosynthesis along with pigments inside of chloroplasts. So we want to talk about some of those guys. So let's start off with sunlight, shall we? Sunlight, as you already know, is a mixture of a whole bunch of different energy. Lots of colors. You already know the colors: Roy, G, Biv, red, orange, yellow, green, blue, indigo, violet. And you probably have heard that the EMR, or electromagnetic radiation, part of the electromagnetic spectrum, is going to include such things as ultraviolet light, this guy right here, which is what actually is going to give you a suntan and or cancer, visible light, which is what you see, infrared, which is basically heat energy that comes from the sun, and you probably heard about some other ones. You've probably heard of X-rays and gamma rays, and beyond infrared, we have something called microwaves, and finally radio waves. And actually, there's ELF waves, which are extra long frequency waves as well. So that's what plants are using to do their thing, and so we want to learn a little bit more about what's going on with those wavelengths of light. So here we have Roy G Biv. It's kind of backwards on this one, but looking at this thing, we can see that all of those numbers that you see there are wavelengths of light. So the longer the wavelength of light, the less energy it has. Which makes sense because when you think about it, the longer the wavelength, the less often a wavelength is going to crash into something that it's hitting. The more often a wavelength crashes into something it's hitting, the more energy it's going to have. And since light always travels at the same speed, the longer the wavelength, the less often those things will crash. So we look at something like this. Red is going to be the thing that has the least amount of energy, whereas a violet's going to have the most. No doubt you've looked at a flame before. When you look at a flame, you'll see that a flame is going to be at its hottest. Probably you learn this at the blue portion of the flame. The red portion of the flame is actually going to be have the least amount of energy. So as you look at those are some numbers that we're going to look at a little bit when we talk about what types of wavelengths things are either absorbed or emitting when they are being exposed to light. A pigment, by definition, means it is something that either absorbs or reflects light. So for example, I happen to be eating M&Ms right now because I am a pig. This is yellow. And what that means is this particular M&M is reflecting all of the yellow light that's hitting it and absorbing all the other colors. Right? This is a green M&M. And a green M&M is going to be reflecting all the green light that hits it and absorbing the rest, much like a plant. So when we look at a plant, like you see in the picture there, plants are absorbing all the other colors of light, or most of them, except for green. So if we had a plant sitting in a room and we exposed it only to green light, chances are plant's not going to live very long because of that. So the two major types of pigments that we have to talk about are going to be chlorophylls and carotenoids. So the first one that we have here is chlorophyll, and there are two types of chlorophyll. There's something called chlorophyll A and chlorophyll B. When you look at chlorophyll A and B, you can see that they absorb different wavelengths of light. They're very close to each other, but they're not exactly the same. And that's actually a really good thing we'll talk about later. So chlorophylls are going to absorb all these wavelengths of light, but they're not going to absorb green light, and that's why they appear green. And that's why most things that do photosynthesis appear green, is because they have just a whole crap load of chloroplasts that are in there. Okay, and they're going to give off that light. And we'll talk about this whole diagram here in a second. So, in terms of measuring light absorption, we'll actually get to use this guy in class, or if you've already done it, you've seen this before, and it's called a spectrophotometer. And a spectrophotometer is going to measure a pigment's ability to absorb various wavelengths of light. Really what it does is it gives you a number that tells you whether or not light is transmitted very well through a sample. So you get a very small test tube called a cuvette that will put a sample in there. We'll put it in the machine, we'll be looking at specific wavelengths, we'll hit a button, we'll look to see what the transmittance is, and if we look at that, if I had something like a cuvette filled with chlorophyll solution, which I know is green, which means it reflects green light, if I were to take white light and divide it up into all the colors and take just the green light, and that green light's going to go through right here, I know that green light is not going to be absorbed by chlorophyll because I know chlorophyll reflects it. So any of this green light that comes into the cuvette is going to reflect the green light out of the solution into my spectrophotometer, and I'm going to see basically 100% or very close to that transmittance because none of that light is going to be absorbed by the chlorophyll. On the other hand, if I had something like this blue M&M here, okay, now look at my next picture. If I could switch that so that blue light was being exposed to this chlorophyll solution, I know that chlorophyll absorbs all colors except for green, and it reflects green. Well, if that's true, then when blue light hits it, it will be absorbed by the chlorophyll solution. And if it is absorbed, it never makes it through the cuvette, and we'll find out that over here this transmittance will be very low because very little light is going to be getting through there. So ultimately we're going to use this method to find out what colors, well, specifically we want to find out what wavelength things are being absorbed at, so we can compare those to some pigments that are found in plants. Conclusion time. What does this all mean? Why do we care? Why we care and what it means is right about here. This is what we really want to focus on right here. Okay. Since we know that green light is reflected by chlorophyll, we must then agree and understand that photosynthesis is going to take place the best when it is absorbing other colors of light. So specifically, it turns out for chlorophylls A and B, it turns out that violet, blue, and red tend to be the types of light that actually work best for photosynthesis. So not green light, but those other colors. Those are the colors of light that are actually going to help, we'll find out later, move electrons around on the thylakoid membrane and help us to produce more ATP. So if you take a look here, we have an absorption spectra that has 3 different chemicals on it. One of them is chlorophyll A, one of the chlorophylls, another one chlorophyll B, and then finally this other class of things called carotenoids that are going to absorb different wavelengths of light. And you can see they're all very similar to each other, but they're all just different enough to be specialized for a different wavelength of light. So chlorophyll A, you can see down here, absorbs something in this range, and we'll find a number for that later. Whereas chlorophyll B, right here, is going to be this guy. He's absorbing somewhere up in this region, so they're slightly different. We also have those same 2 curves up here, 2 other wavelengths of light that we're going to be focusing on, that guy and this guy. Finally, we have this other class that's in yellow, and those are called carotenoids, and you can see that it's a slightly different color. There are a few other ones that absorb pretty well up here in the red and orange range, and that has a lot to do with why leaves change color in the fall, and I think we had talked about that earlier in the year. So in the summertime, when chlorophyll is working great, most plants are going to look green. And of course what happens is, as the year progresses and the amount of light that is exposed to the plants by the day dwindles, we start to see that chlorophyll begins to shut down and it begins to denature. It's a protein. And when that happens, it turns out some of these other chemicals that are in the leaf start to become, well, they're not start, they are actually active at the same time, but they continue to act for a longer period of time than chlorophyll. So because the chloroplast, or excuse me, because the chlorophyll is denaturing, that color that is normally reflected is no longer reflected as well, and so we start to see these other colors, which of course are the colors of light that these other pigments do not use. They reflect them, and so they use slightly different colors. And that makes sense because that means that a plant is basically covered in terms of using many different wavelengths of light to do photosynthesis. It's not dependent on just one wavelength of light, and that's actually a really good thing. So all of these chemicals we're talking about, all these pigments happen to be located inside the chloroplast of the plant cell. And specifically inside the chloroplast, there are these stacks of things called thylakoids. And you can kind of see them in this diagram down here. In my mind, they look like little green hockey pucks that are stacked one on top of each other. Each thylakoid is surrounded by a membrane, and that membrane we call a thylakoid membrane. When there's a big stack of these things together, a whole bunch of thylakoids, that has another name, which is confusing, but it's called a grana. So when you look down here, this whole structure right here, that's called a granum. Each one of these things that's inside here that it looks like a little hockey puck, those are each going to be individual thylakoids, which are surrounded by the thylakoid membrane. The reason why this is also important is that later on when we get into the real aspects of what's going on in photosynthesis, that membrane is going to be really important in terms of creating ATP and creating sugar. So we want to make sure we know the structure of that thing before we go any further. So your leaves, which of course are the major sun collecting organs of the plant, are basically there, and they do a couple things. They absorb sunlight, but they're also going to have to take in carbon dioxide, because that carbon dioxide's got to get into the plant someplace, because that's going to be one of the main things we need to start the whole process. So it turns out, and we'll look at these in class, but if you look on the underside of a leaf and you magnify things very carefully, you can actually see there are these little guys down here, and they look almost like little doors. There's one right here, and you can see in the middle there's a line. That line right in the middle, these are 2 cells called guard cells, and those two guard cells can actually sort of open up like this, and when they open up, that will let carbon dioxide in. Unfortunately, it also lets water out, so that's a problem. But it turns out that when those things open, we can take in some carbon dioxide from the air, get it into the leaf cells, and we can do photosynthesis. And if those things don't open, we can have a problem. So at the end of the unit, we're going to talk a little bit about two other types of plants. Most plants are what we call C3 plants, and then there are two other kinds that have to do with this strange carbon dioxide situation. They're either called C4 plants, or we call them CAM plants, CAM, which stands for a much longer term. But it turns out these cells that are here, called guard cells, have a really important job of letting carbon dioxide into the leaf, and that's where all the gases are going to go in, and then oxygen can dissolve out through those things. Okay. So the last thing is when you look at a chloroplast, which we've learned about, we know that they do photosynthesis. This chloroplast is this entire guy right here. Okay. The chloroplast has its own membrane. Within that, there's yet another set of membranes, which we'd mentioned a second ago, and those are called thylakoid membranes. And so you have these places that are sort of the hockey pucks, the thylakoids, that are inside those things, and there's outside those things. So you can either be in this membranous space inside here, which is not inside a thylakoid, but still inside the chloroplast, or you can be inside the thylakoid. And that's important because that means we have these two different areas, which means we can set up different concentration gradient, which is going to be important. Well, all the fluid that is outside in between the thylakoids we call the stroma. Sorry about the weird name, just get used to it. And then within the other things, we'll call that the thylakoid, and we'll call it the thylakoid membrane. Okay. Beyond that, that's really all you need to know for this now. At this point, we're going to be stepping forward and looking at what's going on within the thylakoid, and that's quite complex, so I'll take my time as I go through that. And if you need to watch that one more than once, please do. Otherwise, we should be all set and ready to talk about the inner workings of photosynthesis. Thanks.